All right. (8 a.m. Friday morning. And I can't remember where we left off. So I think we left off on grain. Is where I think we left off. Is that right? We didn't do time yet. No. This is the last one? All right, so yeah, just to kind of review what we're dealing with is this intraspecific variation. So variation in genotypes or phenotypes across a species. What causes that? And we talked about the spatial factors, or we're talking about the spatial factors and the temporal factors. So spatial factors, number one is that magnitude of difference between our patches. So the stronger the difference, the more, the bigger the magnitude, the more unlike those habitats are, the stronger our selection pressures, and the more variation we'll see across that species. That's kind of modulated by the ecotone, which is that barrier, or that boundary layer, because if the boundary layer is gradual and our species can live in that area, then it kind of just homogenizes all of our variation. Because yeah, you have one extreme over here and one extreme over here, but you've got the middle ground right in the middle of that boundary layer, of that ecotone. So if it's gradual versus abrupt, that can have an effect on our variation. Isolation between our patches. So the more isolated you are, the less likely you'll have gene flow coming in to homogenize our population. So isolated groups, isolated habitats tend to go down their own evolutionary path that adds the variation to our species. And then the third spatial factor is this idea of grain, where grain relates to our patch sizes relative to organismal mobility. All right, so interesting thing, I walked out of the garage today, and there's a hawk that had a had a dove. I know it was one of those pigeons out in my grass. So I saw it and I stopped and I backed up into the garage and set everything down so I could get a picture, and by that time he flew off. But yeah, feathers all over. I mean, that hawk sees its patch, sees its environment as very fine-grained. It sees a lot of detail, sees the forest, the different types of forest, sees the shrublands and all of that stuff. All right, so fine-grained environments, organisms going to see and experience different types of habitats, which means evolution is going to try to balance all of its phenotypes. That results in a decrease in intraspecific variation. But if a species sees its habitat as being very coarse, meaning that it probably only ever sees and experiences one habitat patch, then you have the possibility where each patch is pushing its populations down their own evolutionary path. They're causing divergence, which in the end causes more intraspecific variation. Temporal variation also has a role in this intraspecific variation. And the reason it does is because our environmental variation can change from year to year, from day to day, and so forth. All right? And anytime it changes our fitness, changes our selection pressures. Now, the thing with the temporal variation is that it's going to vary on different scales, it's going to vary in speed, and it's going to vary in predictability. So on the scales type of things, we have variation that occurs daily. All right? Warm temperatures during the day, cooler temperatures at night. But we also have seasonal variation. So average daytime highs are going to be warmer over the summer than they are over the winter. And also with the seasons, the range of temperatures. So in summer, we don't really have a huge range of temperatures between night and day. But once we go into the winter, now because we have a continental climate, we can see upwards of 40 to 50 degree temperature swings, daily temperature swings. We also have the magnitude of change. So how big is that change seasonally? In places in maritime climates, the summer to winter temperature differences really isn't a whole lot, but in a more continental climate, those magnitudes could be much higher. Also, the farther north you get. So where I grew up outside of Chicago, our summertime highs, 80s and 90s. We were up in the 90s. I tend to forget it as a kid because it seemed like running around as a kid in 90-degree heat was just hot. You were sweating, and then that's what you did. But I know it was daytime highs can get up into the 90s. But then over the winter, you're talking daytime highs, teens and 20s at times. All right, I missed some high school. Days were canceled because our daytime highs were projected into the single digits. That's a very large temperature swing, seasonal temperature swing. It could vary in speed, so seasonal changes are actually pretty slow. Right? We can see it coming, all right, and then our daytime highs gradually increase as we go into the summer, and then they gradually decrease. We're on our downward decrease right now. All right, but other times it's very fast, and those are the daily swings. So this morning it's going to heat up, and it's going to heat up fairly quickly. As well as predictability. That's the other thing. So we know daytime swings are predictable. It's going to happen every day. We might not know how high it gets or how cold it gets, but it's a predictable change. And same thing with seasonal changes. Now, how does this all affect our intraspecific variation? Well, it affects the speed and the ease in which our organisms can adapt. So our organisms are going to better adapt to predictable changes, to slow, gradual changes, and things that allow evolution to act. All right? If it happens quickly, meaning within a generation, our organism really can't respond. It really can't respond. So how does that affect our intraspecific variation? Well, you have to think about how our organism responds. Is it going to be responding to these genetically? And if so, then you have the possibility of increasing your variation. Or is this going to be adapting more through acclimation or phenotypic plasticity? All right, so recall. Species. These are groups of individuals, groups of populations that can interbreed and produce fertile offspring. Within these species, you can have subspecies, or at least a finer level classification. These subspecies are local, phenotypically distinct forms. So we've classified them as subspecies because we can see the differences. All right? All of our classification has all been on visual differences. All right? What can we see that allows us to classify and separate those organisms? Subspecies could also be called a race. All right? And a lot of times, these subspecies, these races, are based on geographical ranges. So you can have a species that extends all throughout the United States, but then they can take on different phenotypes because of where they live. So then naturally we say, well, we've got a subspecies here in the southwest of the United States. We have a subspecies in the northern plains. We have a subspecies along the east coast and so forth. All right? When we have that, when we have those species or those subspecies, that tends to be linked to the genetics. All right? It seems to be linked to genetics, which leads to this intraspecific variation. Now, our phenotypic variation, an example that I have is this color polymorphism in these snails, sepia snails. This is just kind of a range of it. These are all the same species. They can all interbreed. They can produce fertile offspring. But they're found in different geographic locations. Those locations vary on their predation pressures. So certain areas had higher predation. Other areas had less predation. In those areas that had high predation, there is a much closer match between the shell pattern and color on these snails and their background on where they're normally found. So you can kind of rationalize that this color, this background matching, is all about camouflage, hiding from predation pressure. And since predators lived in different areas, all right, that kind of explains your geographic patterns. We're matching what we experience in the environment. Now, this pattern matching is oftentimes due to the genetics, the underlying genetic code, your genotypes, meaning this is nature. All right, nature type of variation. But it doesn't always have to be. It can actually be linked to phenotypic plasticity, which is that nurture component. So our snails or our other organism that we're interested in senses the environment, senses the predation pressure, and then matches the color or matches tolerance limits so that they can survive and they can reproduce. An example of this, or if we deal with the nature, it leads us to the formation of ecotypes. So before we get to the phenotypic plasticity part, we'll start with ecotypes. So what are the ecotypes? So recall how we defined ecotypes. These are genetically distinct populations that are adapted to their local environment. Our subspecies, you can pretty much classify them as ecotypes because they are usually genetically distinct. They're also phenotypically distinct. But just because you're genetically distinct, just because you're an ecotype doesn't necessarily mean that you're a subspecies, that you're phenotypically distinct. So we've got that kind of separation. We can be both genetically and phenotypically distinct. We can be genetically distinct only. We can be phenotypically distinct only. All right? Issues with that. So we're going to look at these ecotypes, genetically distinct populations. We go out there, we look at our species, we look at the individuals. They all look to be the same, but once we start doing genetic analysis, we see, no, this group here, genetically distinct. It's forming its own group. Our population over here, completely separate. All right? Example of these ecotypes are plants that live in serpentine soils or in areas around the serpentine soils. So what are serpentine soils? They're soils that are kind of harsh to live in. All right? They're classified as having soils that are high in magnesium. Magnesium is toxic to the plants. All right? So what these plants have learned is they've developed a biochemical adaptation that has allowed them to utilize magnesium, which is a 2+ cation, in place of calcium. So they've kind of learned to use it. In some cases, they've learned to take it up and then sequester it away so that it doesn't harm the rest of the plants. All right, so these serpentine soils are high in magnesium. Plants have to learn how to deal with it. These soils are going to be low in nutrients, so low in nitrogen and phosphorus and potassium. You know, those 3 key nutrients for plants. These soils also tend to have very poor water holding capacity. So we're going to see adaptations, we're going to see traits that are similar to plants living in arid environments. All right, now in these serpentine soils, sometimes you have contiguous branches of these soils, sometimes we have patches. So in an area, we can mark off, let's say, a 1-acre part of land, and what we see is these patches of serpentine soils surrounded by non-serpentine soils. But when we look at our plants are found all throughout these regions. Good soils, bad soils, and so forth. So all these X's might be locations where the plants are growing. And if we do a genetic analysis, we'll see that we actually have a lot of variation here. What was that variation? Well, we've got all of those plants that live in the serpentine soils are one genotype. They're genetically distinct, and those that do not live in that soil are another type of genotype. This is the ecotypes. We've got an ecotype that can live in serpentine soils. We have an ecotype that cannot and can only live in those better types of soils. So what caused the formation of these ecotypes? Well, there's 3 things. The organismal factor is that we have to have variation. We have to have this heritable variation. These plants had to have some sort of adaptation in their genes. Maybe it was even just in a heterozygote. Maybe it's a recessive allele. A couple of them had it. So when their offspring found this habitat, selection can increase that trait. All right? So this is an organismal factor. Our organism had to have the heritable variation in order for selection to start making ourselves genetically distinct, or making that group genetically distinct. Fundamental Fisher's theorem, Fisher's fundamental theorem is probably the better phrase. Fisher's fundamental theorem says that the rate at which the mean fitness of a population increases by natural selection is equal to the additive genetic variation in fitness. I'm going to change that. Let me change this. I don't know how. Get it in the correct order. And I'm on the crappy keyboard. Sorry. Oh, Fisher's fundamental theorem. There we go. So what does this mean? Well, it's tied to that heritable variation. We talked about the rate of selection, the rate at which we see the changes, and it was tied to this heritable variation. This is Fisher's fundamental theorem. If you have traits that are coded for by the genetics, and these traits are heritable, meaning they're additive, then we're going to see a very rapid change. So the more that these genotypes, the more that we have the traits that allow our organism to survive those serpentine soils, the more we have that, then the faster we're going to evolve these ecotypes. All right? So that's our organismal factor. The environmental factors are related to the intensity of selection. So that goes back to, previous lectures again. The evolutionary ecology. Intensity of selection. The stronger the selective force, the faster the rate of change, if we have that heritable variation. All right? Serpentine soils. These are pretty severe. If you don't have the traits, you can't survive. All right? Definitely, if you don't have the trait to deal with that magnesium, you won't survive. That is a very high selective force. But if we're just talking about, let's say, a good soil and a slightly not so good soil, well, those selective pressures aren't very high. The differences aren't very high. So our ecotype formation is going to happen very, very slowly, if it happens at all. And our last factor, an environmental factor, is the barriers to gene flow. All right? We can't have organisms moving back and forth because that's going to break down the forces of selection. Remember, gene flow homogenizes allele frequencies. It can oppose selection. So if we have evolution pushing groups down their evolutionary path, gene flow can eliminate that, can get us back to the same starting point. In this case, yeah, our plants can move back and forth, but the soils are so harsh, the selection pressures are so high, that even if these guys, the ones that can survive in serpentine soils, get back into the good soils, those adaptations came at a cost. They're not as fit as the rest of them. They're going to be outcompeted, or I should say, the non-adapted groups will outcompete all of the ones that can live in serpentine soils. All right, so genetically, the nature component, we can form these ecotypes. Ecotypes are going to be favored when we have things like very harsh environments that are predictable, that are slow, that allows our organism to adapt. But in other times, when it's unpredictable, when we don't know where we're going to get, oftentimes the best solution is to evolve a mechanism of phenotypic plasticity. So recall, phenotypic plasticity is the ability of our organism to produce different phenotypes in different environments. And we've talked about this before. So this is, organism is raised, it senses its environment, and it changes its phenotype. Doesn't change its genetics, doesn't have enough time to do that, but it's able to change its phenotype. And by changing its phenotype, it changes its fitness. All right? So our phenotypic plasticity example that we talked about, remember, is sun leaves versus shade leaves. I know I talked about it that first week in lab. We said in the sun leaves, we don't need as much surface area in our leaves because they're in full sun. They can capture plenty of light. And that means when we look at these leaves, we have very deep lobes. All right? And the leaf captures more than enough light to not only sustain itself, but to contribute extra energy to the tree. Shade leaves, though, they don't get a whole lot of light. So their lobes are much shallower. Our surface area of the leaf is going to be much bigger. And it's all about capturing the light. Now, in these trees, they sense where they exist as those leaves are growing. So you can't take a sun leaf, put it in the shade, and expect it to change. You can't put a shade leaf in the sun and expect it to change. All right, that's for the trees. But in some cases, we do see that. Marine iguanas are one, where they change their phenotype throughout their life. This isn't a process of them growing and maturing. This is an actual physical change. Change in the density of their bones. Yeah, go ahead.
Sorry.
Oh, that's okay. That's okay. They change the density of their bones. They change their muscle mass. And why do they do that? Well, they do that because smaller individuals are more energy efficient. And they would do this when they are nutrient deprived, when they're in an environment that doesn't have a whole lot of nutrients to begin with, or their food supply is low. When does that happen? When does that happen? It happens during El Nino years. All right, so our marine iguanas are near the Galapagos Islands. You can go back to our last presentation in the aquatic environment, and we talked about how we have those upwellings that happen along the western coast of South America. Those upwellings brought nutrients to the surface. You had a whole lot of primary production, including the algae, the red and green algae that is the food source of these iguanas. All right, and that algae, that primary productivity, leads to a boom in diversity in that area. Well, in the El Nino years, we have a shift in those currents, in those oceanic currents. And what happens then is you don't get the upwelling in that area. And if you don't get the upwelling, you don't get the nutrients. If you don't get the nutrients, you don't get the abundance of this red and green algae that's a food source. So during these El Nino years, these marine iguanas, you can say they sense, or perhaps they're starved, they're not getting enough food. So in response, they have a decrease in overall body size. Their bones shrink, their connective tissues shrink, they shrink as their phenotype. When El Nino goes away and the upwellings return, now these marine iguanas increase in size again. All right, this is an example of phenotypic plasticity throughout the life of our organism. They change it. Now we had a question, would an octopus changing its texture to blend in count as plasticity? It could. It could. We'd probably note that they have that ability to change colors if we were trying to classify it as a colors. So phenotypic plasticity is really just changing of our phenotypes. All right. Any questions? So what are the advantages of phenotypic plasticity? Well, the obvious advantage is that it can adapt to its environment and it can adapt quickly. All right? It's not going to be stuck with a single phenotype like our plants here in these serpentine soils. They've evolved the phenotype to survive those serpentine soils. They can't get rid of that. That is baked into their genetic code. But with phenotypic plasticity, it doesn't matter because you have one genetic code and you have the ability of producing more than one phenotype. So, if we have a situation where phenotypic plasticity is favored, then those individuals that can respond are going to be more fit than those that cannot respond. And thus, phenotypic plasticity, the ability to be phenotypically plastic, becomes favored. This is much more prevalent in variable environments. Our organism doesn't know where it will be, doesn't know what type of environment it'll be in, it doesn't know when it'll change. In those conditions, phenotypic plasticity will be favored. You can produce more than one phenotype from a single genotype. The downside of this, though, is that it costs a lot of energy. It costs a lot of energy. And the energy that we're really talking about is the regulatory mechanisms that have to be in place. It's not only about being able to change your phenotype. It's also about sensing the environment. and then knowing once that environment changes what to do. Those pathways cost energy to keep up, to maintain. All right, so if you compare an organism that can be plastic and an organism that isn't plastic, this one that isn't plastic costs less energy. Now, the ability to change could way exceed that cost of not being plastic. So, yeah, it's energetically expensive. And that's why I said here, if our environments become constant, then the ability to be plastic is lost. That fitness benefit is lost. You don't need to have a different phenotype. You produce one phenotype and that's it. So take out the serpentine soils and say, we've just got a great soil habitat. Yeah, it doesn't matter. Why be plastic? Evolution will get us to our best fit phenotype, or could get us there. So in dealing with phenotypic plasticity, we, as ecologists, population geneticists, can make a graph called the reaction norm. This gives us the range of phenotypes that we can see for a specific genotype across a range of environmental conditions. Now, the importance of this is that it'll tell us how the genotypes can respond, because some genotypes, no matter what you have, they're all going to respond the same way if our organism can be phenotypically plastic. In some cases, they're not plastic and they respond the same way. So that's what these graphs show. So if we have no phenotypic plasticity, then if we look at the different genotypes, and each of these lines represents a different genotype, then our trait, our phenotype, is the same regardless of our environment. Across our range, we have the same phenotype. Now, the phenotypes are different, and that's due to the genetics, to our genotypes. Our genotypes are what produced our different phenotypes. If we have phenotypic plasticity, then what we see is the phenotype changes as our environmental level changes. So the trait itself changes. Let's say we have the trait that we're interested in is height at maturity of a plant. All right? So genotype 1 in the red line grows to a high mature, it's tall when it's mature. And it's tall no matter what environment it's grown in. All right? So that has no plasticity. But if we have a plant that can exhibit phenotypic plasticity, well, that's a tall plant to begin with. But if we change our environment, you can see that our height at maturity is actually lower as we change our environmental conditions. In this case, our decrease in height across those three genotypes is basically the same. So, yeah, we have a genetic difference, genetic differences leading to the difference in height, but we also have the effect of phenotypic plasticity. What we can also have is not only a difference in our genotype, not only a difference of our phenotype, but now we have what's called an interaction effect. So how we respond, how our phenotypically plastic trait responds, depends on our genotype itself. So some of it, in the red line, actually decrease when we change our environment. But on these other genotypes, they increase. This I don't know, pinkish one increased in its height, and the bluish one, maybe it didn't change much at all. All right? And this is kind of interesting because the best case scenarios are you have this or you have this. That's our best case scenarios. Very easy to test, very easy to separate. Worst case scenario is you have something like this, and then if you encounter that, then you go to your research advisor and say, I think I want to pick a different project. And we don't deal with it because the complexity is a little bit harder. All right. Now, why do we present this? Well, the reaction norms, you can see it. This is something that you can make. Make a project and say, I'm interested in phenotypic plasticity. So one of the first things you're going to do is make these reaction norms to see, is it entirely genetic? Is it entirely, or is it, does it have a phenotypic plasticity component? And if it does have phenotypic plasticity component and a genetic component, are they interacting in one of these strange ways that will produce drastically different responses? Now, how does this apply to intraspecific variation? This is an FYI only. This is something that we cover in our graduate level evolutionary ecology class. When we look at genetic variation, you have basically the same amount of variation across all environment levels. All right? So if we go out and sample a population, whatever genetic similarity or genetic differences we get, that can be reliable. If we go to a site like here, this has much more variation than this. And that variation is due to the environmental effects. So we might think about populations and say, oh, we've got much more variability up here than we do here, and you'd say, oh, that must be genetic variation, but it's not. It's actually due to phenotypic plasticity, and we have to correct for it. And then here, it looks like we have a lot of genetic variation, a lot of phenotypic variation across the range of things, and it's actually due to both genetics and phenotypic plasticity. So if you're interested in this sort of thing, you can take evolutionary ecology, learn about some of this. If you go on to a graduate school someplace else, you'll probably encounter this in either like an evolutionary ecology course or perhaps a population genetics course. But know that it's not just genetics, it's not just phenotypic plasticity. You can have things react very, very strangely. So, summary between our genetic adaptation and phenotypic plasticity. Which one's favored under which conditions? Well, if we have a constant environment, we're going to have very little variation. Very little variation in our environment, very little variation in our selection pressures, and that means that we should favor a genetic response and not phenotypic plasticity. Why is that? Why would we favor a genetic response if we have a constant environment? And this constant environment deals with both spatial variation and temporal variation. If you're constant, why should a genetic response be favored? Take a guess. So we're favoring genetic instead of a phenotypically plastic response. Okay, so yeah, it's longer, it lasts longer, and that allows evolution to act. That allows evolution to act. And the other thing to think about is also that if we're constant, there's really no benefit in changing a phenotype, having multiple phenotypes with one genotype. There's no benefit. It doesn't change. You're under the exact same selection pressures. So anytime we have these constant environments, constant spatial or temporal, we're allowing genetics to take over. We're allowing evolution to kind of push us down that best fit phenotype. Fine-grained environment. So these are the environments where our organism sees multiple types of habitats. And with this fine-grained environment, it's possible that our offspring are in a different environment than which we grew up in. All right? So that means that our organism, our species, could encounter a lot of variation. And because of this, we favor phenotypic plasticity. All right? This is kind of like the flip side of that constant environment. All right? Evolution is going to have a very hard time producing our best fit phenotype because it doesn't know where we're going to fall. It doesn't know where our offspring are going to land. So in the good soils, evolution's pushing us one way. In the bad soils, evolution might be pushing us in a completely opposite way. All right, now the serpentine soils is kind of its own situation, but we can kind of get, let's say, sun versus shade, light gaps in a forest. Should we grow tall or should we grow short? Well, you have one genotype, and it's the phenotypic plasticity that allows our plants to grow tall or to grow short. All right? We can have more than one phenotype from a single genotype. And that's going to be good anytime we have these variable environments. And especially if we don't know where our offspring are going to fall, where our offspring will live. And then our third case, temporal variation, if it's very rapid and if it's very unpredictable. Phenotypic plasticity is going to be favored. Why is that? Yep. Yeah, so because of the constant change, I have in my notes, same explanation for this fine-grained environment. All right, this temporal variation, if it's unpredictable, we don't know when conditions are going to change. We don't know what our new conditions will be in the future. So it benefits us to be able to change our phenotypes. Marine iguanas, it doesn't know, it can't predict when an El Nino year is going to happen. All right, it doesn't have, I should say, we don't think it has the cognitive ability to actually rationalize that stuff, right? All right, we don't want to anthropomorphize those. All right, they don't know it, it's unpredictable. But from a single genotype, it has the ability to produce a more energy efficient phenotype to survive those low nutrient conditions. As far as the speed, if we have this rapid change, we don't have enough time for evolution to act. So, but phenotypic plasticity can respond pretty quickly. So even among our offspring will sense that we're in a different environment, they'll produce the phenotype that it does better there. I also have the reverse applies to each of the above, so just kind of think about variable environments. Right? It's going to be the opposite of constant, and we're going to have the opposite. We'll favor our phenotypic plasticity. Coarse-grained environments, what are those? Well, those are basically our constant environments. They'll favor genetic and so forth. All right. So, genetic variation. Hopefully, you can already understand the benefits of having this genetic variation in a changing world, in an unpredictable world. The more genetic variation we have, the more likely it is that we have that variation that allows us to survive if the environment changes, if we find ourselves in a different habitat, different area. All right? That goes back to Fisher's fundamental theorem. All right? We have to have that heritable variation. And how much of that heritable variation we have determines how quickly we can respond. So what are the factors that increase genetic variation? What are the factors that decrease genetic variation? This is going to be kind of a quick review. We've already talked about this stuff, most of this stuff in the evolutionary ecology, as we talked about Hardy-Weinberg and stuff. So genetic drift, that causes a decrease in genetic variation. Why is that? So we have fixation of alleles. Genetic drift will remove alleles from our population if given enough time. Natural selection. Not all forms of natural selection will cause a decrease in genetic variation. Disruptive selection actually causes an increase. But directional selection and stabilizing selection can cause a decrease. Especially stabilizing selection, we have that compression of our phenotypes. All right? Inbreeding, something new. We had mentioned it, but inbreeding can cause a decrease in genetic variation. So what is inbreeding? It's mating among related individuals. I'm sure you talked about this in introductory biology. We measure the level of inbreeding using the inbreeding coefficient, F. I'm going to warn you, we'll see F again. All right, so the inbreeding coefficient is the probability that two alleles are identical by descent. So if we drew, a family tree, right, we're looking at two alleles that our offspring has, and we want to know how likely is it that we got those from family members, that we got them both from a relative. All right. Large inbreeding coefficient means that we have a high similarity, that it's a high likelihood. You look at, I think the genetics class uses British monarchs, maybe, as examples of inbreeding, where like cousins mate with cousins, right? And that's kind of why some of those family lines had some genetic diseases, heritable diseases that kind of wasn't too good, all right? So why does inbreeding remove Genetic variation, it removes this heterozygotes. It removes heterozygotes, which is going to produce our homozygous individuals, and by doing that, it's going to expose these recessive alleles to selection. All right, so just the act of producing homozygotes isn't really going to be enough to decrease the genetic variation. We're going to need that selection to remove those recessive alleles. All right? So at least genetic drift and natural selection has appeared before. We've talked about that. Factors that increase genetic variation. Again, most of these we've talked about. Mutations. It's the ultimate source of genetic variation. Mutations is what produces those new variations. Sexual reproduction. We talked about this. I know I talked about this on our nature walk. I don't know if Dr. Skipper did or not, but on our sexual reproduction, recombination generates new variation, right? Because with diploid organisms, you have possibility of recombination producing new sets of gametes. All right? This is great when we have changing conditions because that adds that variation on which selection could act. We have to have that variation present if our organism is going to respond. All right? The other thing about sexual reproduction is this idea of the dominant and recessive alleles, just the basis of having dominance. All right? Dominance present is going to contribute to the variation. And I have C heterozygote refuge for that because that kind of is what caused us to think about it. Selection can't remove a lethal allele from the population. All right? And the reason why is that lethal allele is going to hide in that heterozygote. So we need selection to get us close, and then we need something else. We need drift, we need gene flow. Disruptive selection. That's going to promote genetic variation because it's going to help us maintain two phenotypes. All right? Stabilizing selection kind of compressed us. Disruptive selection is kind of separating us. A lot of times people think that disruptive selection may be a way that starts to push us towards our own species classification. We'll leave that for evolution to kind of contend with that, talk about that process, but know that disruptive selection adds to genetic variation. And the new thing that we're going to talk about is frequency-dependent selection. So what is frequency-dependent selection? Frequency-dependent selection is a form of selection in which the fitness of a gene is determined by its frequency in our population. All right? So I have this graph here. This figure is in the book minus the red line. All right, so I added this red line to show you what happens when we don't have frequency-dependent selection. When we don't have frequency-dependent selection, the fitness of our genotype is the same no matter what our frequency is. So this is kind of what we've been working with. When we've said, yeah, we have this allele that is lethal, or that causes a 20% reduction. The selection coefficient is, let's say, 0.2. We've said that that selection coefficient remains 0.2 no matter what, and that causes our change. We're going to gradually increase our frequency of that dominant allele. All right? If our environment changes, then that selection coefficient should change, but we've assumed that it doesn't. This, frequency-dependent selection, is the case where it does change. Now, depending on how common or rare our phenotype is, our genotype is in that population, that's going to determine our fitness. Usually, what happens is if we're rare, we have a fitness advantage. So as we become less common, our fitness increases. As we become more common, our fitness decreases. All right? And I have this benefits of being unique to kind of emphasize that. So this frequency-dependent selection is going to happen when our fitnesses change based on the frequency in the environment. This is going to promote our variation. All right? Example, quick example, fruit flies. All right? Fruit flies. Fruit flies can have red-eyed, red-colored eyes. That is our wild type condition. All right. Or it can have wide-eyed. That's going to be our mutant condition. In our fruit flies, there's a study that looked at the frequency of eye color across time. So what they did was they started with an almost complete vial of red-eyed flies and introduced wide-eyed, real low frequency of wide-eyed. And what they ended up seeing was that our frequency increases. And our increased up and then stabilized at a certain frequency. All right, in that frequency, it was about 40%. So it stabilized at about 40% wide-eyed flies in that vial. I said, oh, that's interesting. So 60% red, 40% white. They then took the vials and said, okay, well, what happens if we go above that 40%? Well, if they went above the 40%, the frequency of the wide-eyed flies decreased until it got back to 40%. This is frequency-dependent selection. Wide eyes has higher fitness, and that caused a decrease. That caused the increase in wide-eyed. In this zone, the fitness of the wide-eyed flies is less than the fitness of the red-eyed flies, and that caused our decrease. This is frequency-dependent selection. Why did that happen? It came down to mating. When you were rare, that one wide-eyed fly obtained more matings per fly compared to the red-eyed flies. So yeah, you may have 10 red-eyed flies in there, and each of them could have mated once, but that one wide-eyed fly probably mated 3, 4, 5 times. That wide-eyed fly had an advantage. It mated more, and that meant that it had more offspring and got us to the 40%. Once we exceeded that, now the female flies are looking at it and say, oh, yeah, I've seen you before. I'm not going to mate with you. So it goes for the red-eyed flies. All right? And that wide-eyed flies suffer. We drop down to 40%. There is something, just for completeness, 80%. At 80%, we shot up to 100% wide-eyed flies. You can say this is frequency-dependent selection because the fitness of the wide-eyed flies exceeded that of the red, but it's for a different reason. This was about mate selection, competition for mates. This was actually due to competition. So the more wide-eyed fly larvae that were in there that were growing up in the median, those actually outcompeted all of the red-eyed larvae and drove us up to fixation. What about these lines? This is where our fitnesses were equal. That's where our fitnesses were equal. All right? So frequency-dependent selection, it's going to maintain those two different phenotypes. It's going to promote our variation. And this is a very common occurrence that helps to maintain all of this variation in our populations. All right? Any questions? What we'll do is we'll wrap up with a review and with the summary. And we will talk about how to quantify and how to measure some of this variation. How do we distinguish between phenotypic variation or phenotypic plasticity and our genetics? We're going to finish this stuff on Monday, and that's going to be the end of our exam material. So anything that we talk about on Wednesday is going to be for that next exam. All right? I believe this is chapter 7 in the book. So we skipped chapter 6. We're going to talk about some of that chapter 6 stuff later on. But if you have the textbook and you're interested in reading, this is going to be our chapter 7. Yep. Next week, Friday, a week from today. So start looking at it. I will have a quiz on this and a quiz on the second presentation. What I might do is go ahead and finish this quiz on this presentation and get it posted, but it's not going to be due until, I think, by the end of the day on Tuesday. So that way I can release it on Wednesday, and you'll have Wednesday and Thursday to review the keys and the answers and stuff. All right? All right. Enjoy the weekend.